Hi guys, we're doing Specimen paper 2022, paper 1 for biology, 9700 variant 01. Okay, so from 2022 onwards, you're gonna have 1 hour 15 minutes. 15 minutes have been added to the paper. Cambridge basically wanted consistency between all the papers, right? So now you have 1 hour 15 minutes for all three science papers: physics, chemistry, and bio. So let's begin with question number 1. Number 1, which definition of magnification of a drawing of a leaf is correct? Remember, the forms of magnification is m is equal to image length by actual length. M is equal to I by A. So the actual size multiplied by the magnification of the microscope. That's wrong. The measured difference in size between the leaf and the drawing? No. D looks like the correct answer. The size of the drawing of the leaf, the image length, divided by the actual size of the leaf. Okay, so the size of the drawing image divided by the actual size. So one should be D. All right. Number 2, the electron micrograph shows part of two cells. Which labeled features identify these cells as eukaryotic? Okay. So as you can see, W is actually a chloroplast. This is a plant cell. W is the chloroplast. X is the cell wall. So basically, cell walls are also present in bacteria. Do you understand? So X is not a definitive feature. Could I clarify? So X shouldn't be the answer. So we are only left with C, which is W. Also, Y looks like the cell membrane, which is also present in the cell surface membrane is also present in prokaryotes. So 2 should be C, right? The chloroplast. It is a eukaryotic characteristic. Okay. Moving on to 3 now. Plant cells are stained and viewed using a student microscope. The light source is natural light. What could be clearly visible at 400 x 400 magnification? Basically, we cannot see ribosomes. It's 20 nanometers in diameter, right? So we cannot see ribosomes. We cannot see cristae. We cannot see the grana of chloroplasts, right? We hardly see the mitochondria. How are we gonna see the details of mitochondria, right? Because it's like 7 nanometers, the cristae, the diameter, the thickness, right? So we can see nucleoli though. They're gonna be stained properly. So 3 is C. Uh, Wait a bit. Yeah. So for 4, which lengths are equivalent to 1 micrometer? Let's see. 1 micrometer is basically 1 into 10 to the power minus 6 meters. Okay. Remember that. It's like this. This is meter, millimeter, micrometers, and nanometers. This is 10 to the power 0, 10 to the power minus 3, 10 to the power minus 6, and 10 to the power minus 9. So 1000 millimeters is actually 1 meter, not 1 micrometer. So 1 is wrong. 0.001 nanometer. Nanometer is smaller than micrometer, right? By 10 to the power minus 3. So if you want to go from smaller to bigger, smaller to bigger, this is basically 1 into 10 power minus 3 nanometers, right? If you convert it to millimeters, sorry, micrometers, it's going to be 10 to the power minus 6 micrometers. So 2 is also wrong. Okay, let's go to 3. 0.001 or 1 into 10 power minus 3 millimeters. If you convert this to micrometer, multiplied by 10 to the power plus 3, so you're going to get 1 micrometer. 3 is a correct answer. So 4, this is 10 to the power 6 nanometers or 10 to the power 3 micrometers. So 4 is not correct. 0.01 millimeter, that's 1 into 10 power minus 2 millimeters or 1 into 10 power minus 2 into 10 power 3, which is 10 micrometers. So 5 is not correct. 1000 nanometers, 1000 into 10 to the power minus 3 micrometers, that is equal to 1 micrometer. So 6 is correct. So it's 3 and 6, right? 4 is D. 5. Some secretory cells synthesize and release glycoproteins. What is the correct sequence of some of the events that occur in a secretory cell? So guys, exocytosis will occur at the end. Okay, one should be at the end. It's releasing the contents. So at first we need mRNA to bind to ribosomes. Okay, so 3 will occur first. Then we're going to produce the proteins or the glycoproteins, right? Then we're going to pack them into secretory vesicles. That will be done by the Golgi body 3, and then this will occur. So it's actually 3 4 1 2 3 4 3 products accumulate in secretory vesicles. Okay, so about this, like I said that it's 3 4 1 2, right? Sorry, 3 4 2 1. Yeah, 3 followed by 4 followed by 2, and at last we'll have exocytosis. So 5 is B. Okay. Number 6, a scientist used a blender to break open a sample of animal cells and release the organelles. The extracted mixture was spun in a centrifuge at low speed. The heaviest type of organelles sank to the bottom of the centrifuge tube to form a solid pellet, pellet 1. The liquid above pellet 1 was poured into a clean centrifuge tube and spun at a higher speed. The next heaviest organelles sank to the bottom to form a solid pellet 2. So basically the logic is, in a centrifuge tube, the most dense or the heaviest pellet will sink to the bottom at first. Okay, and after you repeat the procedure, you're gonna get them one by one. All right, so let's try to understand the process. They want pellet 3, so let's classify them according to the sequence. Let's do this. So what's the heaviest here? Let's identify them first. Synthesis of proteins, this is actually ribosomes. Okay, digestion of old cell organelles, this is actually lysosomes, right? Synthesis of mRNA, this is the nucleolus. Production of ATP by respiration, this is actually the mitochondrion. Understood? Synthesis of mRNA, that happens in the nucleus. Basically, the nucleolus is in charge of making nucleolus is in charge of making rRNA, and mRNA is synthesized in the nucleus in general. That's the organelle. All right, so the heaviest one is the nucleus, followed by mitochondrion, followed by lysosome, followed by ribosome. So our answer should be A. 6 is A. It comes in the third spot, right? Hopefully that makes sense. Now what? What is the general formula for amylose? Amylose is basically the polysaccharide. It's a component of starch. It's actually unbranched. It only has 1,4 alpha 1,4 glycosidic bonds. Okay, so we're gonna have a one less molecule of water. It's like this. This is typically for glucose, right? Now if you subtract one molecule of water, what do you get? C6H10O5, right? C6H10O5, and this will be multiplied by N. That's the simplest general formula for polymers, polysaccharides. 8. Which statement describes how the molecular structure of starch is suited to its function? Starch is basically a storage carbohydrate for storage. Amylose has a branch structure, and amylopectin is called to give a compact molecule for transport. It's not mainly used for transport. In the breakdown of amylose and amylopectin, many condensation reactions release stored energy. No. Hydrolysis reactions take place in breakdown. In the formation of amylose, many hydrolysis reactions allow. No. Many condensation reactions. Formation is condensation. D. Amylose and amylopectin form a complex that is insoluble, right? So it does not affect the water potential of the cell. Guys, this is the most important part about starch and glycogen. They are insoluble, okay, in water, so they don't actually affect water potential. That's why we can store them. Number 9. Cows and whales are mammals that produce milk to feed their babies. Newborn whales grow faster than newborn cows. Milk contains fatty acids with a range of chain lengths. The table shows the percentage of fatty acids of different chain lengths in cow milk and whale milk. Okay. Which statement about the ratio of short fatty acids to long fatty acids, 18 or more carbons, in the milk of cows and whales is correct? 9A. The ratio in cow milk is higher because newborn cows need more energy than newborn whales. That doesn't make any sense. The ratio in cow milk is lower. Okay, so basically the ratio of short to long, okay, it's higher in cows. Basically, look, cows have 22.2 percentage of short chains. So the ratio in cow milk cannot be lower. Okay, so B is wrong, and the ratio in whale cannot be higher. Okay, we can either say that the ratio is higher in cow milk or the ratio is lower in whale milk. Do you understand? Hopefully that makes sense. So A doesn't make sense. What about D? The ratio in whale milk is lower because newborn whales need more energy than newborn cows. Okay, so think about it. Whales are in general, look at this, newborn whales grow faster than newborn cows. They're obviously bigger in size, right? So they need more energy. Now, obviously, longer chain fatty acids can provide more energy because they have more CH bonds, which can be hydrolyzed to provide energy using ATP, right? The hydrogen can be released, right? The CH bond can be broken down. So remember that the longer the fatty acid, the more energy it can provide. So since whales have a higher percentage of longer fatty acid chains or a lower percentage of shorter fatty acid chains, this is a result of newborn whales needing more energy than newborn cows. Cows don't need that advantage. Okay, that's why the answer is D. 10. The structure of phospholipids includes a polar phosphate head. That is true. Hydrophobic fatty acid chains. That's true. And glycerol, saturated fatty acid chains. We're not sure about that. It may contain unsaturated fatty acids. We aren't sure about 3. Okay, 3 makes no sense, so 10 is A. 11. The diagram shows the molecular structure of a peptide. Which molecules would result from complete hydrolysis? Okay, so let's break down the amide bonds. Okay, we're gonna break this amide bond and this one over here. Okay, so what compound are we getting here? This is the compound that we're getting. This CO will be converted to a COOH, right? Carboxylic acid. What about the one in the middle? This is going to turn into NH2, right? NH2 followed by this, and this is going to turn into COH. And this one will be COH, CH, CH3, NH2. Pretty simple, straightforward, valid, right? So let's see. Basically, if you look at the compounds over here, how are you going to identify the correct ones, right? So let's take this step by step, then we can understand there is what would result from complete hydrolysis. What's wrong with A? This group is extra. This shouldn't exist, okay? What's wrong with B actually? Look at this. This part is wrong. It should have been CH2OH, right? They've given this compound. I don't know why. Let's go to C now. 11 looks like C. C is perfect. Okay, what about D? This part is wrong. It should have been CH3, okay? 12. The diagram shows a hemoglobin molecule. As you can see, we can see the alpha helices properly, right? Hemoglobin is a globular protein composed of 2 alpha and 2 beta globin chains, which identifies different parts of the molecule. Okay, let's see. 3 is clearly an alpha helix. You guys need to understand that, okay? 3 is an alpha helix. So 3 should be an alpha helix. So clearly, since they've labeled the alpha helix in 3, A and D are excluded, okay? So what about the other parts, okay? So hear me out. As you can see, 1 is actually pointing to a particular compound over here, something that doesn't look like a protein, okay? it could have been both of these. It might be the heme group or the oxygen binding site because it's the same thing. This is the heme group or the oxygen binding site. So the difference is actually between 2 and 4. Look, 2 actually refers to a region which is facing the exterior part of the compound, and 4 refers to a region which represents the interior part of the compound. Since this is a globular protein, remember that the hydrophobic amino acids will point towards the center. So 4 actually refers to the hydrophobic amino acids, and 2, the external part, refers to the hydrophilic amino acids. So 12 is B. Get it? 13. A student investigated the effect of enzyme concentration on the rate of hydrolysis of protein in milk. When the enzyme and milk were mixed, the protein was hydrolyzed and the mixture changed from cloudy to clear. Okay. The student investigated 5 different enzyme concentrations and recorded the time taken for the mixture to become clear. What is an appropriate control for this investigation? So what is the control used for? Basically showing us that the purpose of the experiment is valid. Basically saying that the enzyme is required for the reaction. Like saying that if we didn't use the enzyme, the reaction wouldn't occur in the first place. Do you understand? So if we use something instead of the enzyme, the solution wouldn't turn from cloudy to clear. It would remain cloudy. So what can we use instead of the enzyme? We can use distilled water. So 13 is A. Get it? That's the purpose of a control. 14. What determines the specificity of an enzyme? So remember that specificity is mainly determined by the tertiary structure of the protein. That means the 4 types of bonding present between the R groups of the polypeptide. So 14 is mainly 1. These things, okay, optimum pH, covalent bonds between, this is the primary structure. Primary structure doesn't determine specificity. The shape of the substrate molecule, okay, this indirectly depends on the tertiary structure. So one thing determines the specificity of the enzyme, the shape of the active site. It's basically the tertiary structure or the bonds between the R groups of the amino acid. Okay, so 14 is C, only 1, the tertiary structure. 15. The graph shows the effect of temperature on the rate at which the enzyme in a biological washing powder digests and removes fruit juice stains. Which statement explains the shape of the graph at temperatures higher than T? So basically denaturation has occurred, right? Bonds are broken between R groups. So one looks good, one is fine. There are more collisions between the enzyme and the substrate. No, then rate would increase, right? So 2 is wrong. Clearly 2 is wrong. So the answer has to be C. The tertiary structure is altered, the shape of the active set and the substrate are no longer complementary. 16. Which statement describes carrier proteins in a cell surface membrane? They are found on the outer surface of the membranes, allowing cell recognition. That's not the function of a carrier protein. That's the function of a receptor protein. Carrier proteins are involved in moving substances through the membranes by either active transport or facilitated diffusion, passive transport. B is true, 100% true. Okay, they are mainly involved in transport. Okay, and carrier usually can do both, and channel proteins can only take part in passive transport, facilitated diffusion, but carrier proteins can perform both. 17. What happens to a typical bacterium when it is placed in surroundings that have a higher water potential? So water will come in and it won't burst. Why? Because the bacteria actually has a cell wall. Okay, but look at this. What happens to a typical bacterium when it is placed in surroundings that have a higher water potential than the water potential inside the cell? 17, right? The bacterium bursts because the cell wall has no structural function. The bacterium dies because water leaves the cell by osmosis. Water cannot leave the cell. Okay, the bacterium would not burst because the cell wall would protect it. There is no change because the cell wall is impermeable to water. That is false. There is a neat movement of water into the bacterium. This is true. Okay, A is wrong. 18. By which processes do hydrogen carbonate ions leave the red blood cells? Okay, so remember, carbonic anhydrase catalyzes this reaction between H2O and CO2 to form H2CO3, which dissociates to form H plus and the bicarbonate anion. So since there is a huge excess of bicarbonate inside the cell, right, it will come out of the cell by a process called facilitated diffusion because it's not active. Okay, it's a passive process. Remember that. 19. In an experiment, fluorescent dyes were attached to proteins on the outer surface of cell surface membranes. Fluorescent dyes of one color were attached to proteins of a living human cell, and fluorescent dyes of a different color were attached to proteins of a living mouse cell. The human cell and mouse cell were then fused to form a hybrid cell. At first, the proteins attached to different fluorescent dyes remained separate, but after 40 minutes, the proteins were distributed randomly across the hybrid cell surface membrane. What does this experiment show? It actually tells us about the fluid nature of the membrane, right? Proteins are found only on the outer surface of the cell membrane. No. Proteins in the outer layer of the bilayer do not penetrate into the inner layer. Proteins move freely, right? That's it. That's the fluid nature, the fluid mosaic model, right, of the cell membrane, the phosphated bilayer. They can interchange their places, okay? So yeah, the proteins can move freely in the phospholipids of a cell because typically the ones from the mouse and the humans were separate at one point, but after one point we got some from the mouse here, some from humans, mouse here, humans here, so they interchange their positions, right? 20. The diagram shows the mitotic cell cycle during which phase is DNA replicated. Since this is cytokinesis, right? This is the, you know, mitotic, out of the mitotic cell cycle, this is cell division and D is nuclear division, right? Nuclear division. So this whole part is interphase. This is G1, this is S phase, and this is G2 phase. So DNA is replicated during the S phase. So the answer should be B, 20B. 21. Bacterial cells divide by a process called binary fission. Which macromolecules must be synthesized for binary fission? Basically, DNA is replicated or they divide by binary fission mainly, okay? So cellulose is totally wrong because bacteria do not have cellulose. 3 is wrong, okay? So the answer must be B, 1 and 2. So we need DNA and peptidoglycan because peptidoglycan is part of their cell wall, and cell membrane proteins and RNA is required, obviously, okay? 22. The electron micrograph shows a group of human chromosomes. Which label is correct for W, X, and Y? So W is basically the centromere, okay? W is basically the centromere. X is at the end of a chromatid. X is basically, what is it? X represents the telomere, right? And what about Y? Y represents the whole thing, right? So it represents one of the parts of a chromosome, that is one chromatid. So 22 should logically be C. Moving on to 23. Which statement about the behavior of chromosomes during mitosis is correct? They attach to the spindle fibers to contain them within the nucleus. Not really. They condense to prevent further translation of genes. Not really. They reach the poles of the cell and begin to uncoil. Okay, that's true. This happens after telophase, right? After they have reached the poles. They actually attach to the spindle fibers to, line them up along the equator, right? They condense to prevent further translation of genes. It should have been transcription of genes. They replicate to produce sufficient DNA to form two nuclei. Sufficient DNA. Double, I think it should be double. Okay. It shouldn't be sufficient maybe. So C is the best answer basically, right? 24. Gene mutations can occur by substitution, deletion, or insertion. What is the smallest part of a DNA molecule that can be changed by gene mutation? Okay, so remember, you can change. The smallest part is a nucleotide actually. Okay, it's a nucleotide. Codon represents 3 bases. Gene is a section of DNA. Nucleotide is one of them. Okay, a nucleotide is one of them. What is a base? A nitrogenous base is either, pyrimidines or purines, right? But we don't say that the base changes. We say that there is a change in the nucleotide. It comes as a unit. It has 3 parts, right? The deoxyribose or ribose sugar, deoxyribose for DNA, phosphate, and the base. So we say that the nucleotide is the smallest part that can be changed. Which statements about mRNA are correct? mRNA can form base pairs. That is true. We do see base pairs in mRNA. They can form hydrogen bonds. Obviously, if you form base pairs, you form hydrogen bonds. It is single stranded. So all 3 are correct. That's true. Moving on to 26. The table shows the role of 4 different proteins involved in DNA replication, which shows the functions of these proteins. Okay. Helicase adds DNA nucleotides to the 3' end of a growing nucleotide strand. Mm, about this, this is not right. Okay, this is not right. It's basically required for unwinding the DNA, making double-stranded single-stranded. Okay. DNA polymerase is required for synthesis. Topoisomerase breaks and rejoins the DNA strands, right? And single strand SSBP, single strand binding protein, binds to separated DNA strands to stabilize them so that they don't come back again together, okay? So the best answer here clearly is D. Why? I'll tell you. Helicase actually makes strands available as templates. Okay, and topoisomers, remember this, they actually, it enables tension caused by unwinding to be released. So it basically breaks and rejoins the DNA strands. So remember that unwinding and topoisomers, like helicase and topoisomers, have different roles. Helicase is mainly in charge of breaking the hydrogen bonds, okay, making double strand DNA single stranded, and topoisomerase is required to actually, remember that there's some, when you coil two things up, remember there's tension between them, because if you let it go, remember it's gonna, just think of two coils and you're wrapping them around, okay, in a coil, right? Two coils, you're winding them around in the shape of a helix. If you let it go, they will have a tendency to open up by themselves, right? Some tension is stored. So topoisomerase actually enables tension caused by unwinding to be released. That's the role, remember that. DNA polymerase adds DNA nucleotides. This is the role of DNA polymerase. Okay, it should have been here. That way you could actually eliminate A and C, right? And SSBP, single strand binding protein, it prevents original strands reforming. Okay, it stabilizes. Like after you, um, uncoil parts of DNA like this, some of it will be still bound like this. SSBP prevents reformation or rewinding. Okay, you need to remember this. This is a very important MCQ 26. Okay, single stranded binding protein prevents the original strands from reforming. Okay, number 27, which type of sugar and which type of bond are found in a DNA molecule? Okay, so in DNA we have deoxyribose sugar. It's a monosaccharide actually. Okay, so it's a monosaccharide, and what bond is formed in a DNA molecule? So we have hydrogen bonds between the two strands. Okay, so 27D. 28, these are transverse sections of 3 plant diagrams, the root, stem, and leaf. So this is very important, guys. Remember, we only deal with dicots, right? So in one, remember this, never forget this. The answer for this should be C. I'll tell you why. Phloem, we're looking for phloem, all right? So remember the star-shaped region is actually xylem, okay? And this section over here, this is actually the phloem. And for stem, the outer section is actually the phloem. Could I clarify? And the inner section, the one pointing towards the center, is actually the xylem. What about the leaf? What about the leaf? 6 is actually phloem. The one in the lower part of the vascular bundle, that's the phloem. Okay, hopefully you remember. Just remember one, you can easily remember the other one that way. Okay, really important question. 29, sucrose moves from phloem sieve tube element into a root cell, which changes to the water potential and the volume of liquid in the phloem sieve tube element are correct. Okay, so this is the root, this is the sieve tube element. So after sucrose moves from here to here, what happens to the water potential of the sieve tube element? It loses some solids, right? So water potential must become higher. But the water potential of this decreases, water potential of this increases. Now what will happen after sucrose moves in? Water will flow from sieve tube element to the root like this, right? So the volume of liquid in the sieve tube element will decrease. 29A. 30. Which statements about water movement in plants are correct? Water can pass through cell walls containing layers of cellulose fibers. That is 100% true. The apoplast pathway, right? Water can pass through cell walls containing rings or spirals of lignin. That is also true. Water can pass through suberin. That's 100% right. Okay, because remember the Casparian strip, the apoplast block pathway is blocked, but water can pass through spirals of lignin in xylem, some xylem vessels. Okay, so all three are correct here. Moving on to the last section of the video, we are seeing a xerophyte, right? It has some adaptations. Which label features are characteristic adaptations of many xerophytes? So 31, the correct answer should be C, right? Why though? 1 basically refers to the sunken stomata. So water particles, water vapor is stored here. This actually reduces the steepness of the, you know, water potential gradient. 2, these refer to some hairs. They also, um, trap air. It prevents the flow of air, so that actually reduces the steepness of the gradient. So 1 and 2 are fine. 4 is the vascular bundle, and 3 are actually air spaces. Those don't actually help, right? And 5, what about 5? The waxy cuticle and multiple layers of upper epidermis. Typically, in the plants that we see, we only see one layer of upper epidermis, but here we're seeing multiple layers. That's the benefit, okay? 32. A maize crop is growing successfully in a field in which the water potential of the soil is minus 40 kilopascals. What is the most likely water potential of the cell sap in the root hair cell? So it is growing properly, right? So water is coming from the soil into the root hair cell, okay? So for this to happen, this must be more positive than the root hair cell, so this must be more negative compared to minus 40. The only possible answer is minus 60, or else water wouldn't come in to the root hair cell, right? Because the soil is at minus 40. For water to move down a potential gradient, it needs to go from minus 40 to minus 60. Okay, 32 is A. 33. The contraction of the heart is coordinated through electrical impulses passing through cardiac muscle. What is the correct order of impulses? The sinoatrial node creates the impulse, okay? Then the atria will contract, followed by the AVN, which delays the impulse, then Purkinje tissue. 33 is B. 34. Which features allow the aorta to withstand high pressure? Remember, it's mainly the elastic. It is an elastic artery, so it should be the elastic fibers. You're going to look for elastic first, okay? Now what? Elastic fibers are endothelium. Endothelium doesn't really have a structural role in withstanding pressure. It's going to be collagen. Okay, 35. Which way is correct for pulmonary arteries? Pulmonary arteries, remember, they carry blood, deoxygenated blood actually. It's the only artery in the body which carries deoxygenated blood from the heart to the lungs. Okay, so since it's an artery, this is an exception. Since it's an artery, the lumen size will be small, okay, and the thickness of the wall will be very thick comparatively. 35D. 36. The graph shows oxygen dissociation curves of adult hemoglobin in different carbon dioxide concentrations 1 and 2. Percentage saturation of hemoglobin with oxygen. Which conditions could change the shape of curve 1 to shape of curve 2? Okay, so we're seeing the Bohr shift. When does this happen? When carbon dioxide concentration increases, okay, remember, CO2 binds with H2O to form H2CO3, which dissociates to form H plus and HCO3 minus. Since H plus is formed, pH actually decreases. So the answer is A, 36A. Remember, guys. 37. What maintains the necessary concentration gradients for carbon dioxide and oxygen in lungs? By the way, guys, some things have been removed, sorry, from the syllabus. We don't have the effects of smoking, tar, nicotine, emphysema, bronchitis, COPD. Those have been removed. Okay. Also, one other thing, some diseases have been removed. We only have malaria, HIV, TB, and cholera now. Only these. No measles or smallpox. You do need to know about eradication though. 37. 37 was the best answer. What maintains the necessary concentration gradients for carbon dioxide and oxygen in the lungs? Good ventilation of the lungs. This is the correct answer. Okay. This maintains the gradient because oxygen is coming in and carbon dioxide is going out frequently. This helps in diffusion, right? These also help in, these three help in diffusion, not maintaining the gradient. Which cells and tissues are present in trachea? Trachea have goblet cells. They also have cartilage. They also have ciliated epithelium. So 38 needs to be B. The global mortality figures of some diseases in 2013 are shown. How many millions of people died from 2013 from bacterial diseases? Okay, this is a virus. This is bacterial. 1.40. Malaria is caused by Protoctist. Cholera is caused by Vibrio cholerae. Okay, it is also a bacterium. So it should be 1.42 in total, 39B. Why is it difficult for B lymphocytes and T lymphocytes to respond to antigens on pathogens that are intracellular parasites? Okay. Basically, you need to understand the antigens need to be exposed. Okay. to identify them. Now, if they're intracellular, try to understand what happens when an intracellular parasite infects a cell. They won't be detected. They will stay inside our cells. Okay, so the lymphocytes do not even encounter them. They will clearly recognize them as non-self, but they won't even encounter them because they're stuck inside our cells. That means they will be hidden. We need to present them, right? Okay, guys, that's it for the specimen paper 2022. If you have other questions, comment down below, and I'm going to link bio paper 1 up here, and I'm going to link the paper for Feb March 2022, right, variant 1 2 up here, and I'm going to link the variant for Feb March 2021 variant 1 2 down here. All right, and remember to subscribe to the channel if you like the content. See you guys.